Hi everyone, Assalamualaikum, Salam Sejahtera, I'm Sharifah. I am going to be presenting on novel insight into the role of oxidant activation of protein kinase G in resistant artery. So let's take a look at what is resistant artery. It is a small artery that carries a big burden in determining blood pressure because it have to accurately regulate the volume, the pressure, and the flow of oxygenated blood towards the downstream capillary in order to meet the end organ demand. Three distinct features that can only be found in small arteries is that first, it constricts in response to pressure, also we know as myogenic tone. And second, it has a feature called myoendothelial projection, where it is packed with calcium channel. It is known as a microsignaling hub. And third of all, it is highly innervated, and that's why it can dilate and also constrict quickly in response to organ demands. So this is the myoendothelial projection that I talked about. We will be imaging this feature so that I'm showing you so that you know what I'm referring to. And one of the most important mechanism or highly conserved mechanism is the acetylcholine-induced vasodilation. So I just would like to bold that you will see this simplified mechanism throughout the slide. Quickly going through it is acetylcholine. We know that it activates GQ-PCR, and the alpha subunit activates the membrane-bound protein phospholipase C, which cleaves PIP2, giving us a very important messenger that regulates the calcium. So regulation of the calcium that I'm referring to is the localized calcium within the myoendothelial projection region here. Okay. So where does the oxidant, came into the picture? So we got to know that protein kinase G can be activated by oxidant. So we have the classical pathway that's been established for 50 years ago, that PKG activated by CGMP. However, recently we know that PKG can also be activated by oxidant at a region whereby it forms the formation of interprotein disulfide bond between two residue of PKG located at cysteine 42. So in order to make this pathway silent, what we did is that we developed a mice where I'm going to call it knock-in going forward, that cysteine is being changed to serine. So imagine one change of amino acid from cysteine to serine leads to this animal being hypertensive at a very early stage on. So it is confirmed that this animal is hypertensive. So, but how do we know if, got anything to do with the endothelium? Because we know PKG is one of the most abundant protein in the vascular smooth muscle, and its vital role is for dilation in vascular smooth muscle mechanism. So the question is, how does oxidant activation of PKG plays any role in small resistant artery endothelial function? So in order to study this, we have to look into the myogenic tone, like I mentioned before, only distinct in small resistant artery. So here we have to order whereby we pressurize it, and then once we will wait for it to develop myogenic tone. So here you can see that the diameter is decreasing, is getting smaller and smaller. So high diameter, lower number in diameter. So when it reaches plateau, this is when our artery is ready to be challenged by any agonist. So here, first of all, like I said, this mechanism is going to be seen throughout the slides. That the first mechanism would know whether oxidant activation of PKG is involved is to challenge it with ACH. If you can take a look at here, we can see that full dilation of acetylcholine at 10 micromolar in comparison to only 20% of dilation. So it shows that the muscarinic-induced endothelial vasodilation, is very conserved regulatory pathway. We know this. However, we don't know, we never knew that oxidizable PKG is highly crucial, it is critical in this mechanism. So in order to verify this, we bypass the whole mechanism and straight activate the IKSK channel with NS309. Here you can see that they both dilates normally, indicating that there's no abnormalities in the IKSK channel. Hence, we have to dissect the mechanism, upstream. So first, we look into TRPV4 calcium influx by activating directly the TRPV4 channel with GSK101. You can see at 10 millimolar, sorry, nanomolar. Oh my God, at 10 nanomolar, you can see almost a full dilation as compared to calcium-free. However, in the PKG knock-in mice, there's only 20% of dilation. If you can take a look at here. Sorry, here 20% of dilation. However, when we challenge it, to 30 and 100, you can see there's a right pharmacological shift. What does this indicate? This indicates there is a competitive agonist within the endothelium. So in order to scrutinize the mechanism further, we're going to look into IP3 calcium release. However, pressure myography is not really a great method to look into IP3 calcium release. The best method is imaging. So we use a high spinning disc confocal laser microscopy to image. So what we did is we take the same set of arteries and then prepare it on fast, which is endothelium upwards, load it with fluorophore and image it. So one of the key features of this microscope is that it is able to take 60 frames per 1 second. So it allows us to see this transient movement in and out of calcium ion within the different channels and receptor. So this is how it looks like. You can visualize each and every endothelial cells clearly. And the white spots here is the myoendothelial projection. So we are looking at from upwards here, okay? So you can see this is the endothelial and this is my endothelial projection. So calcium pulsar here refers to calcium release. So we name it calcium pulsar. So it pulses because it pulses. So you can see that normally this is normal, but adding 10 micromolar of acetylcholine, pay attention to the activity. It's highly active and the frequency is increasing and there's increase in number of sites. Okay, so now we noticed that at baseline, there was no difference at all between the two. So we look into number of sites, which is different sites, of the IP3 that's been detected, indicating by the colored square. And also the frequency, how many times it releases calcium in the square, in the region of interest here, and also the pulsar amplitude. Please take note that the pulsar amplitude is our positive control because we need to know that we are imaging the right calcium channel, the right receptor. In this case is inositol triphosphate receptor where it releases calcium. So in activation with ACH, what we notice is that here, can you see there's a lot more activity going on compared to here? Only 1, 2, 3, very few activities. So when we analyze the result, you can see that there's a twofold increase in number of sites, which means that there are more receptor is being activated when we add acetylcholine. And within that receptor, there's more frequency. There's even more release of calcium that's been released from the IP3R. You can see the twofold increase. However, like I said, our positive control is the same, which tells us that yes, we are imaging from the right calcium receptor, from the IP3 receptor. So this method can be, we can image from different, receptor from TRPV4 and other calcium channel receptors, calcium channels as well. So conclusion of the findings, what do we know? So this is the mechanism that you're looking at, is the highly conserved mechanism that we know for, 50 years ago. So this is what we see from our current experiment. We see that there's a low calcium entry and calcium release, and there's a low response in TRPV4 activity to its agonist. That's why there's no dilation. So there's three things that needs to take note. No response to ACH, no response to TRPV4 activator, and reduction in calcium release. Hence, it is upstream. What we further postulate is that oxidant activation of PKG might act as a biological anchor, in order to activate phospholipase C, or in order to aid the cleavage of PIP2 by phospholipase C. So now, when there's no biological anchor, because PKG cannot be activated by oxidant, you don't see, so all of this mechanism is silent, hence no dilation at all. So why is this study very important? There's a lot of people still, affected by respiratory hypertension worldwide. But come on, let's focus in Malaysia. There is a remaining large cohort of patients with uncontrolled hypertension, and they are exposed to other comorbidities, which leads to more complex clinical management for this patient. And this patient I'm talking about is 1.5 millions of Malaysians. That's a big number, because 6.4 millions of people has been diagnosed in Malaysia with hypertension, and only half are aware they are hypertensive. And out of this half, only 90% are taking one or combination of this known medication in the market. However, only 45% of these patients who are on medication have their blood pressure level controlled. So that leaves us with 55% of our of 55% of uncontrolled hypertension, which brings us to 1.5 million Malaysians. That is Malaysians only, So obviously, there is an unmet need. It is a dying need to find a new therapy, a new pharmacological agent. So we hope by understanding the physiology, it could lead us to generate a targeted and controlled calcium signaling in arteries that may represent a new therapeutic target that could restore endothelium function in a different way compared to other known medication that's in the market. So here I'm showing one of a recent paper by one of our collaborators that they're trying to find a compound that could activate protein kinase G in the manner that we study. So there's a compound, we call that G1 drug. So this G1 compound are able to attach or to form disulfide bond by 93.7%. So we hope, after toxicity study and after a lot of, preclinical model, that we could actually prove that this compound might work. So 10 years from now, when we know how FDA works, hopefully it is out in the market. So what to take home from my presentation is that M3 muscarinic activation induced endothelial vasodilation is a very highly conserved pathway. It's been there for 50 years. We know this. All of the physiology and in medicine, we know this. However, recently we just found out that oxidizable PKG or PKG that can be activated by oxidant are highly involved in this mechanism. So evidence shows that oxidant activation of PKG is critical in endothelial physiology. So everyone, thank you so much for listening. And these are my friends and colleagues from University of Malaya and also University of Manchester. And these are the authors involved in producing the paper. So thank you so much, everyone. Have a good day.